I'm going to talk today not only about a novel approach to using an existing ArcGIS tool to answer a conservation question about fish, but also how it took a lot of different individuals with many different expertise to complete this project as well. So let me introduce us first. We're a natural history survey research team based out of the Springfield DNR building. The three of us on the left there that contributed to this project are survey employees, and then there's a couple of DNR employees that contributed a lot to this work as well. And most of our research is in support of the Wildlife Action Plan, especially the aquatic portions of that. And during a recent study, we did a comprehensive update of the fish distribution and abundance in the state, where we took large volumes of data. And one of the things we did was map the distribution of all the fish species in the state. And when we looked at those, we started to see groups of individuals or locations, and we asked questions like, are these groups isolated? What are the isolating mechanisms? And then can we actually measure the connectivity of all these different groups that we see? So we had to know two things. First of all, to answer this question, one was where are the fish? So we created maps like these of all the different fish species in the state so we knew where they were located. But then we had to ask what are the barriers in between all of these locations as well. And an obvious one is dams, and we knew the georeference location of many of the major dams in the state, the idea being those are barriers to dispersal. But also large rivers can be barriers to dispersal, especially for stream species, so sometimes we had to take that in consideration. And when you map the fish locations and these barriers together, you might get something like this, where you have points on a map and barriers in between them, and you can start to see some patterns like in this one, where you might see 4 distinct groups of fish in this particular map. Well, we wanted to test this idea, so we used a modified transportation tool in ArcGIS called Network Analyst, and we applied that to stream networks and said, can we measure the connectedness of any 2 points, knowing that there might be barriers in between those points? And so we started with the map like there on the left, and then we wound up, when we ran that through ArcGIS, like what's there on the right, and we call these different areas in which individual populations occur artificial habitat units. And these artificial habitat units can be defined as the network of stream segments in which the population occurs, or, in other words, the available habitat in terms of a stream network for an isolated population. And GIS gives us a visualization that looks something like this. And of course we wanted to test this because at this point it was all theory, so we used Ironcolor shiner, which is a small minnow species, and it has 2 widely distributed populations that you can see there on the map. But in fact, when we run these locations through this Network Analyst tool, it tells us that we have 8 distinct populations, the one that exists there in western Illinois, but 7 more that exist there in eastern Illinois. So this is the output of artificial habitat units that ArcGIS tells us we have. And according to kind of our meta-population theory, the more spatially separated 2 populations are, the more genetically diverse those populations are. So we thought maybe we could test this using genetic analysis. But further than that, the more temporally separated 2 populations are, perhaps the more genetically diverse they are. And so if you look at the pattern of glaciations in Illinois, these 2 populations may have been separated by many thousands of years in addition to be just spatially separated. So we had to collect some genetic samples in order to run tissue analysis to answer whether or not we could use genetics to help with our ArcGIS. We employed the help of some DNR biologists to help us find Ironcolor shiner. We took those samples and we're going to give them to the CAMO lab here at the Natural History Survey. That's the Conservation and Molecular Ecology lab. They will be able to tell us if, in fact, these different populations in these artificial habitat units do actually differentiate from one another so we can test our theories. And, you know, we can use that information to answer some meta-population type of questions, refine this GIS model, and make it even better, and once again, move it beyond this kind of theoretical exercise. And that's a really neat kind of academic exercise, but the question is then how can we use this in a broader management sense? Of course, once we refine the model, we can apply it to a lot of different species, like here in the western sand darter. We can see if these models are actually accurate. But in a much larger sense, we can ask very specific management questions like, when we remove a dam, does ArcGIS tell us that will help us establish connectivity between populations? What's that mean for the spread of aquatic invasive too if we pull out that dam? Well, this model can tell us that. And furthermore, when we lose populations, say in a fish kill or just from natural random events, Can we expect there to be immigration from adjacent populations? What about genetic compatibility amongst all these isolated populations? But this GIS tool should be able to help us answer those questions.